Hello viewers, today we will talk about auxin, the first plant hormone to be discovered and characterized. As you can see that natural auxins is indole 3 acetic acid. There are two more natural versions. One is 4-chloro-indole 3 acetic acid and another is indole 3 butyric acid. There are several synthetic auxins as well. For example, 2,4-dichlorophenoxyacetic acid and 2-methoxy-3,6-dichlorobenzoic acid. Now, in vivo, indole acetic acid is covalently conjugated with other molecules such as beta-D glucose, myoinositol and aspartic acid. Some high molecular indole acetic acid conjugates are also found with glucan and with glycoproteins. Usually, in vivo, this conjugation is broken to release the free indole acetic acid, which then exerts the biological functions. Now, the physiological effects of auxin actually came from the studies of phototropism, that is, the bending of plant towards light. Researchers used to work with grass coleoptiles, which are protective organs that ensheathes very young leaves. Now, when light is given at one side of the coleoptile, then there is a bending of the coleoptile tip towards the direction of light. This is due to the preferential growth of the cells, basically their elongation at the shaded region compared to the illuminated region. Now, when the tip is cut, then there is no bending, or when the tip is covered with an opaque cap, then again there is no bending. From this, it was concluded that light induces formation of a growth stimulus at the coleoptile tip that is transported to the zone which is shaded and causes those cells to elongate. If mica sheet is inserted at the dark side, then there is no bending. Now, mica sheet is water impermeable. When this mica sheet is inserted at the illuminated side, then there is again bending. If we remove the tip and then put a gelatin block which is water permeable in between the tip and the stump, we can see there is bending upon illumination. This suggests that the growth stimulus is probably a water-soluble chemical that is transmitted to the growth zone and causes the bending. Since it is water-soluble, it cannot penetrate mica, but it can flow through the gelatin block. Now, if an illuminated tip is removed and then replaced at one side of the stump, then even in darkness we see bending. This clearly proves that the growth stimulus is a chemical. It is synthesized in presence of light, and then even in absence of light it stays and it can diffuse into certain region and stimulate cell elongation in that region. Now, based on these studies, Wendt in 1926 developed certain experiments that ultimately led to the design of auxin bioassay. He cut quite a few illuminated coleoptile tips and put them on a piece of gelatin. Then after some time, he removed the coleoptile tips and then he cut the gelatin in smaller blocks. Then he put one block on one side of a stump, which resulted in bending in total darkness. This clearly proves that the growth stimulus was present in the tip, it diffused into the gelatin block, and finally from the gelatin block it diffused into that side of the stump. Now, if Wendt used more coleoptile tips, then finally he had more bending. That means this bending is proportional to the amount of growth stimulus present in the gelatin block, which originally came from the tips. Wendt could measure the degree of curvature, and he saw that the degree of curvature is proportional to the number of coleoptile tips present on the gelatin block. Thus, this could be used as a bioassay for Auxin. Here on the left graph, you can see that the degree of curvature shows a positive correlation with the number of coleoptile tips on gelatin. Then the growth stimulus was isolated from the gelatin block and its chemical structure determined, which is indoleacetic acid, as we have discussed already. And people could develop an assay to measure the concentration of indoleacetic acid in this system. The right graph shows that the degree of curvature is also proportional to the concentration of indole acetic acid in the gelatin block. Thereby, this is a good quantitative bioassay for indole acetic acid in biological samples. Then another simple bioassay was developed where thin sections of coleoptiles are suspended in solutions containing indole acetic acid. After some time, these sections showed elongation, and the extent of elongation is proportional to the amount of indole acetic acid present in the solution. This bioassay actually led to the naming of Auxin from the Greek Auxin, meaning to increase or to grow. Now, other than these bioassays, some modern techniques can also be used to quantitate auxins in biological samples, such as mass spectrometry and ELISA. Now, mass spectrometry in conjunction with gas chromatography gives us two informations together. One is the exact structure of the auxin present in the biological sample, and secondly, its precise quantification, even when the level of auxin is very, very low in the biological sample. Now, auxin has several diverse physiological effects. Many of these effects are mediated by polar transport of auxin. Auxin is the only plant hormone known to be transported in a polar manner. Now, we will discuss some of these major physiological effects, and we will follow it up with the applications of synthetic auxins. The mechanism of auxin-mediated cell elongation has been deciphered at the molecular detail. Here, a part of plant cell is shown where you can see the plasma membrane, the cytoplasm, and the cell wall. Some important components are shown, like a proton pump at the plasma membrane and cellulose microfibrils in the cell wall, which are cross-linked by polysaccharide chains. In these regions, two different proteins are of importance. One is expansin and another group of proteins are cell wall loosening enzymes. Now, auxin triggers this process by activating the proton pump on the plasma membrane, which then exports protons to the cell wall using the energy of ATP hydrolysis. This results in the acidification of the cell wall, and that in turn activates the expansion proteins. These expansion proteins then separate the polysaccharide chains from the cellulose microfibrils. This results in more exposure of these polysaccharide chains to the cell wall loosening enzymes. These enzymes then access the polysaccharide chains and cleave them. Consequently, the microfibrils now can slide. As a result, the extensibility of the cell wall increases. Due to this cell wall loosening effect, when water enters the plant cell, the cell can expand or elongate. Now, the polar transport of auxin, which is important for many physiological effects, can be demonstrated experimentally. A hypocotyl section is cut. This is the apical end and this is the basal end. Then two gelatin blocks are placed on the two ends. At the apical end, the block contains radiolabeled indolactic acid and the block at the basal end is empty. After some time, significant amount of radiolabeled indolactic acid could be recovered from the block at the basal end, indicating auxin is flowing from the apical end to the basal end. That is, the flow is basipetal. If we exchange the position of the gelatin blocks, that is, now the empty block is on the apical side and the block with the radiolabeled indolactic acid is at the basal side, then even after some time, almost no radiolabeled indolactic acid could be recovered from the block at the apical end, indicating auxin does not flow from base to tip. That is, acropetal flow of auxin is not possible, at least in stem. Thus, in stem, auxin follows a polar transport and the direction is basipetal. Now, this basipetal flow of auxin in stem has been characterized again in the molecular detail. Now, you can see that if this is cell wall and this is plasma membrane and this is the cytoplasm, then auxin can enter the cell either via diffusion or via an influx carrier protein called Aux1. Now, these can happen on any surface of the plasma membrane because the influx carrier protein Aux1 is present everywhere. But when auxin exits a cell, it only uses a efflux carrier protein called pin protein. Now, interestingly, these pin proteins are exclusively located at the basal side of the plasma membrane. Consequently, auxin can only flow out in this direction, that is, in the basipetal direction, explaining its basipetal flow in the stem. Now, this reporter gene assay clearly indicates role of auxin in differentiation of vascular tissue. This is a section of a young Arabidopsis leaf. The blue stain is due to beta glucuronidase gene or GUS gene, which is used at the reporter gene. Now, this gene is under the influence of an auxin sensitive promoter. Thus, if there is a blue coloration, that means auxin is there present. Now, this deep blue region is a hydathode where auxin is being synthesized. Now, if we follow the movement of the blue color, we can see that auxin is reaching a differentiating and developing vascular strand. Consequently, auxin must be responsible for the development and differentiation of vascular tissue. In this experiment, using a 7-day-old young internode of cucumber, a wound is generated and 0.1% indole acetic acid in lanolin is applied. Lanolin is applied to give it red color so that the flow of auxin can be easily tracked. Now, as you can see here that there is a basipetal flow of auxin to this region where new generation of xylem tissue and its differentiation is clearly visible. Consequently, auxin can induce development and regeneration of xylem tissue around a wound. In tomato, inhibition of polar auxin transport can block leaf formation at the vegetative apical meristem. Now, this polar transport can be blocked, for example, by mutating the gene encoding the pin protein. Now, due to this block of leaf formation, it results in naked stem formation, which looks like a pin with an intact meristem at the tip. Now, if indole acetic acid is applied on such tip, leaf formation is restored. Thus, auxin can induce formation and arrangement of leaves. Another important physiological effect of auxin is apical dominance. As you can see here that the axial buds doesn't grow. This is due to the flow of auxin from the terminal buds to the apical buds where auxin inhibits their growth. Thus, if the terminal buds are removed, the apical dominance will be lifted. Consequently, the axial buds will start growth. Now, if in such a cut tip where the terminal bud is removed, indole acetic acid is applied, then again we see that the growth of the axillary buds are suppressed. Now, this can only be explained if auxin provides some chemical signals that communicate information over long distance. Auxin also can induce the formation of lateral and adventitious roots. In root, the flow of auxin is acropetal, that is in this direction. As we have seen in stem, it is basipetal. Now, due to this acropetal flow, auxin ultimately reaches the tip of the root or the root cap. Now, from the root cap, auxin again flows back in basipetal direction. Now, due to this flow of auxin, priming of pericycle cells happen. These pericycle cells are the initiator of lateral root formation. As we can see that ultimately the pericycle cells causes initiation of the lateral roots. Eventually, it develops to lateral root primordium and finally the lateral root emerges out. Auxin can also promote development of fruit. For example, this strawberry fruit. The so-called strawberry fruit is actually not a true fruit but a swollen receptacle. These so-called seeds, as we see here, they are actually auxins, that is the true fruits. Now, these auxins, they synthesize auxin and that ultimately results in the development of the receptacle. If these Auxins are removed, then there is no supply of auxin. Consequently, the receptacle doesn't develop at all. But if to this underdeveloped receptacle indole acetic acid is sprayed, then this can again develop to a properly swollen receptacle. Consequently, auxin must be playing a vital role in development of fruit. Two of the most important physiological effect of auxin is, one is phototropism, another is gravitropism. If the illumination is overhead, then indole acetic acid is distributed uniformly along the tip. But if the illumination is at one side, then the indole acetic acid molecules, they flow to the shaded side. And in stem, indole acetic acid induces elongation of the cells. Consequently, these cells, they elongate more than the cells at the illuminated side. As a result, the tip bends towards the light source, the so-called phototropism. In the root, the situation is a little bit different. In a vertical root, as we have seen, there is acropetal flow of auxin to the root cap, and from the root cap there is basipetal flow that distributes auxin uniformly along the surface of the roots. But if the root is kept horizontal, then things change. Here, due to the effect of gravity, the flow of auxin is much higher at the lower surface cells of the root. Now, in root, the effect of indole acetic acid is opposite. That is, it does not promote cell elongation. Actually, it inhibits cell elongation. Consequently, these cells at the lower surface of the root, since they get more flow of auxin, their elongation is inhibited. On the other hand, here the cells at the upper surface, since they get much lower supply of auxin, they elongate more. As a result, the root bends towards the direction of gravity, that is the so-called gravitropism. Since indole acetic acid is chemically a simple compound, different labs all over the world, they prepared synthetic auxins. Now, these synthetic auxins have variety of commercial applications. Interestingly, the mechanism of action of these synthetic auxins are still poorly understood, but they are being widely used for several decades. Some early commercial uses include prevention of fruit and leaf drop, promotion of flowering in pineapple, induction of parthenocarpic fruit, that is seedless fruit, thinning of fruit, as well as induction of rooting of cuttings for plant propagation. Now, in the recent days, the major application of synthetic auxins is as herbicides or weed killers. Normally, indole acetic acid within a plant cell is metabolized very quickly, but these synthetic auxins, their rate of metabolism is very, very slow. Consequently, they stay and they can exert their effect for a prolonged period, increasing their efficiency as weed killers. So, today we have discussed different aspects of the biology of auxin. For example, the different types of auxins, the different auxins that are synthesized in vivo, the natural auxins, the different structures of the synthetic auxins, and then we discussed the development of bioassay, which is very important to quantify auxin in biological samples. We discussed the classical methods as well as the modern methods. And finally, we discussed the innumerable physiological roles that auxins play in plant. Lastly, we talked about the commercial applications of synthetic auxins. Now, this is important about auxin is it is a vital hormone for plant. That means plant growth cannot happen in absence of auxin. That means auxin needs to be supplied to the plant throughout the lifetime. There is only one other hormone that is as significant as auxin. This is cytokinin. So, next we will be discussing about cytokinin. Thank you.